Based off a lot of questions that I was answering while running around the room, I feel like a lot of you want clarification on this, right? So for those who don't remember, on the last exam there was a question that gave you the sense strand of DNA, and they wanted what the mRNA strand would look like. So you guys should remember that the sense strand of DNA was your non-template strand. And there's a long way and a short way of answering this. The long way would be, drawing out the complementary DNA strand. Which will be your template. And then transcribing this into mRNA, which would be which would be the mRNA. That's the long way. The short way is remembering what sense strand means. And basically, if you compare the sense strand to your mRNA, what do you notice? They're the same thing, except for one replacement. Everywhere there was a thymine, there is now a uracil. So one of those answer choices was basically the exact same thing as the sense strand of DNA, except every thymine was replaced with a uracil. So that was the way to solve that one. Sorry, I don't know why this one isn't focusing today. Or maybe it's my eyes, I don't know. But I guess you guys can sort of see that. It's all right? Okay. All right, that's the only one I remember that a lot of people were having issues with. Are there other ones anyone wants me to clarify or cover? The gel? You mean something like, well, that wasn't labeled. A, B, C. And I don't remember which line was in which. I think it was maybe like this. Okay. And then there was that. And then there was that. So same as in the notes, you had it set up like this, and I think it was asking what on here would represent someone that was homozygous recessive, meaning had two copies, and we didn't even use those phrases. We used the phrase two copies of lowercase a, meaning they only show this chromosome. And you guys are supposed to remember that lines that show up near the top are longer, like represent longer fragments of DNA, longer proteins. Their longer stuff is near the top of a gel. And stuff near the bottom is shorter proteins and shorter fragments of DNA. Therefore, patient B represents someone who is lowercase a, because their copy of this looks like that. Yes. Yes, why it doesn't work, right? A lot of you did correctly notice that there were two statements that showed the difference between prokaryotic and eukaryotic, but only one of them is the actual reason why it won't work, and that's the following. So let me write down the two statements. So there's a question saying, if you tried to insert, let me make sure I get this in the correct direction. If you try to insert prokaryote stuff into eukaryote, so if you try to insert prokaryote stuff into eukaryote, why won't it work? And you guys narrowed it down to two choices. One, something mentioning the Shine-Delgarno, and something mentioning the the lack of a 5' G cap. So, although both statements are true that prokaryotic mRNA contains the Shine-Delgarno sequence, and then it is also true that the eukaryote won't read this, turns out the eukaryote doesn't look for sequences in order to figure out how to translate mRNA. Eukaryotes only look for, and it is an oversimplification for undergraduate, but eukaryotes look for this thing. And so because prokaryotic mRNA lacks a 5' G cap, that is the reason why inserting prokaryotic mRNA into eukaryotes without any modifications, you would be unable to translate it. Yes?
So how does the eukaryotic small subunit know where to bind?
Eukaryotic small. They use the 5' G cap on regular eukaryotic mRNA as like a signal to bind to it, and then it'll move along it until it finds the start codon, the AUG. It's kind of like how RNA polymerases in transcription bind to DNA and then keep moving until they find a promoter sequence type thing. Was the whatever answer choice was mentioning the 5' G cap? Yeah. Was it? Can't remember exactly what it was asking. It was something like, why can you use rifampin? Why doesn't it target human RNA polymerase? Human RNA polymerase, and it's the answer choice that says that human RNA polymerase and prokaryotic RNA polymerase are different in structure, or something like that. And you can even extend it, and I don't think it was any answer choice that went into more detail, but prokaryotic RNA polymerase is just one of them. Eukaryotic, there's like 3 of them. Yeah, it's just differences in the structure of the polymerases is the reason why you can do that. And that's actually how all medicines and antibiotics are generally supposed to work, is that they are specific for the prokaryotic version of something, and they ignore the eukaryotic version of something. Okay. Other ones anyone can think of? I'm sure you guys enjoyed the extra credit. I'll have fun grading that. I'll try to get everything done by Friday. I know there's only like a few office hours left. I know there's office hours on Friday and next Monday, and I don't know if Hasties has said yet what he's planning on doing during reading days, so you'll have to ask him. But I'll try to have everything done by Friday so that you guys can start reviewing your exam 3 on Friday. Yes? Not yet, no. Let's see. What else? Yeah, so the final exam review is next Wednesday during FEU's Up All Night. I'll send an email out again giving you guys the info, but it's next Wednesday from 5 to 7 in a different room than usual. It's going to be in SO 250, that spaceship building, basically. It'll be in there. For the final exam review, we actually will be limited based off time because someone's using the room after us. So I will try to get through everything in 2 hours. I will also use the SI session on Monday to try to go over some review stuff. I'll use today and Thursday's SI session to finish covering chapter 30. So I'll try to get through everything, but yeah, just a heads up so that you guys can be disappointed now rather than next Wednesday. Someone is using the room after us on Wednesday, so I only have those 2 hours. I can't stay an extra like 30 minutes like I've been doing before. Okay. Other questions about anything? The exam? Anything? All right. So I guess I'll get started on chapter 30 then. So I will not be covering 29B again because we did that before. So I know that wasn't stressed for exam 3, but 29B will be on the final exam. Chapter 30, we get to the final bit of the central dogma that we care about in undergraduate biology, and that is translation. So chapter 30A, you can think of it as it talks about the components of translation, what codons are, anticodons are, tRNA, mRNA, all that stuff. And chapter 30B actually goes over the mechanisms of translation. So we'll start with 30A, where they talk about how do you translate mRNA into something functional, a functional protein. Well, you're going to see a bunch of vocab terms, and you do need to know what each one means. I'll try to break down a couple of them. One interesting question pops up when you talk about translating mRNA into proteins. There are only 4 DNA bases, but there are 20 common amino acids. How could you possibly code 4 bases into 20 different amino acids? And sometimes I will go back to some of these. But we use the phrase, vocab word here, a triplet code to describe the genetic code. It is a triplet code, not a quadruple or duo or whatever. There's a reason why it's exactly 3. And that's because the 4 bases, if you put them in a bunch of different combinations, if you do it at 3 bases at a time, you can get 64 possible combinations. Imagine it was a unicode, meaning you only need one base to describe an amino acid. What's the maximum number of amino acids you could possibly code if you only use one base? You can only code for 4 amino acids, one base for one amino acid. If it was a duo code, meaning you take 2 bases out of the 4 and you make a bunch of different combinations, how many combinations can you make if you only take 2 bases? It's 4 to the power of 2, which would be 16. There are supposed to be 20 amino acids, so that's not enough yet. So the reason why we have a triplet code is because you take 3 bases, you mix them around in every possible combination you could possibly do, and you get 64 combinations, which is plenty. We only needed 20. So when we create 64 combinations, that is, evolutionarily speaking, the length of the code is a triplet code. 3 bases at a time will each code for 1 amino acid in a protein. All right? Sometimes on the exam, he might say, we've discovered a brand new organism with only 10 amino acids. What code do you think that they will have? Well, if it's only 10 amino acids, they probably only need a duo code, a 2 code, right? So it's just trying to, that question's asked not because there is any alien life forms to ask about, it's asking you, do you understand why we have a triplet code? All right, other vocab terms, let me go back, make sure I'm not missing anything. I'll go over tRNA in a bit. I guess I'll go over tRNA now. Okay, so how do we go from mRNA into protein? Probably the most important hybrid molecule that will help us with this is actually tRNA, transfer RNA. Because on this structure, and you see it's a nice T shape, this structure is a hybrid because on this end right here, the 3' tail end, it is holding on to an amino acid, one of the 20 amino acids that we know about, right? One of the 20 amino acids will be on the tail end right here, attached to that sequence CCA. Down here, we have a region on tRNA that is 3 bases long called the anticodon. Notice it is 3 bases long. This part, think of it as it's like the adapter. This part is going to have some base sequence, 3 bases long, that will match to some 3 bases on mRNA. And if it matches with 3 bases on mRNA, it will dump off its amino acid onto the ribosome, and the ribosome will start building a protein with that amino acid. So tRNA is that hybrid, is that connection between the mRNA level of expression and the protein level of expression. And that's because of this anticodon and stuff up there. Yes? The anticodon part, these 3 down here, and we'll see some diagrams that show it, but if you want to see where it is right now, it's those 3 bases down here. The bottom loop there is what needs to match with the codon in mRNA. There's a bunch of other crazy stuff in here, some of which we'll talk about in another slide when they show some more detail. But for now, this is a general structure of tRNA. Also, interestingly, we have a special nitrogenous base that you get in tRNA that you're not going to get in DNA called, I pronounce it as inosine. It might be pronounced other ways, I don't know. But it's a special base, usually indicated with the single letter I, instead of A's, T's, G's, or C's. It's indicated with I. And you will see that this base actually ends up being fairly important for a process called wobble base pairing, which we'll get to later. But this is the first mention of the phrase, so I wanted to highlight it. It's a special base found in tRNA that will be very important in terms of wobble base pairing, which will be talked about later. Other stuff, some history stuff. We don't care about history stuff. All right, back to this slide. We already went over what triplet code means, but there's some other phrases like overlapping and punctuated, which I believe this slide describes what those mean. Okay. So, vocab terms for what the genetic code is like. 1, it is a non-overlapping genetic code. Here's what this means. Right here, think of it as an mRNA sequence. It's an mRNA sequence. Each color represents, each circle represents a nucleotide in the mRNA sequence. Earlier we said that the genetic code was a triplet code, so notice down here we're under non-overlapping, they highlight every 3 bases, right? That's because they're going to say somewhere in the text that these three bases will code for one amino acid, then the next three bases code for the second amino acid, so on and so on. The phrase non-overlapping code means you do not reuse any of the bases in the preceding codon. So notice up here, which is the incorrect model, overlapping code looks like the following. You take the first three bases to code for the first amino acid, but then you shift over one, you reuse two of those bases, and you bring in a third new base to code for the second amino acid. That's an incorrect model. That is not how our genetic code works. Our genetic code is not an overlapping code. Our genetic code is this one down here, non-overlapping. We use the three bases at a time, and we use them only once. So that's why we have to shift over a whole three bases to get three new bases before we can code the second amino acid. That's what non-overlapping code means, and that's the next vocabulary term you need to know about the genetic code. The other vocab term correctly associated with the genetic code is the phrase continuous code. Okay, so punctuated is incorrect. Continuous is the correct one. So what does continuous mean? Well, let's look at the wrong one. Punctuated code implies that the way the genetic code works is that you use three bases to code for the first amino acid. Then there's like this skip base indicated by like a punctuation, a base that's kind of like a period at the end of the first codon. Then you go look at the next three ones and you use those to code for the next amino acid, another punctuation where you skip, and then you code and so on and so on. That is incorrect. We use every single base in the mRNA after the start codon. Therefore, the correct vocab word is a continuous code. We use three bases here. We use three bases here, and then we move on to the next three bases without skipping anything, and then move on to the next three bases, and so on and so on. So, vocab words so far: triplet code, non-overlapping code, and continuous code. There's going to be more vocab terms in a bit, but that's so far what we've covered. Let me go back again, make sure I didn't skip anything. This slide, by the way, is detailing why we don't have a duo code. You can look at it, but bottom line is if you have a duo code, you only have 16 combinations, which is not enough to code for 20 amino acids. All right, what's this one? The nature of the genetic code. Yep, we already went over these. Non-overlapping, it is continuous, meaning there isn't any punctuation. And now we have another vocab term. The genetic code is a degenerate code. Okay, the genetic code is the degenerate code. Basically what this means, and I'll have to go down to this table here. This table is, I actually don't know what the name of it is, but it's a codon table is what I call it. Basically what this is notice you have on the left here every three base combination that you can get. These are 64 listed combinations on here, every three base combination. Next to it is the known amino acid that is matched to each of these codons. Obviously you don't have to memorize this code either for this class or for the MCAT. A table like this would be given to you. And if you were asked anything about translation or the genetic code or how to translate something, you'd be given this table and given a sequence, and you may be asked, what amino acid sequence do you get from this mRNA sequence? And then you go look at this code, you look every three bases one at a time and figure out what amino acid chain you'll have. So, what does the degenerate code mean? Well, you have 64 possible combinations, but only 20 amino acids. What ends up happening is that, notice, especially for this amino acid, remember, anyone tell me what amino acid this is? Leucine. There are 6 different codons, 6 different combinations that all code for the same amino acid. This repetition, where multiple codons code for the same single amino acid, is what the phrase degenerate code means. Is that there are multiple codons coding for the same amino acid. That is what the phrase degenerate code means. Notice that's true for a lot of these amino acids, like phenylalanine, there's 2 different codons. Histidine, there is 2 different ones. Glycine, there's 4 different ones. It's not true for all of them, though. The one very big example here is this one down here. Methionine is only coded by a single codon, which will be emphasized more in chapter 30b, but anyone can tell me, what is AUG? That is something called the start codon. You will learn in 30b that every single amino acid sequence will start with methionine or a modified version of methionine, because every single codon sequence will start with AUG. Everything that actually codes for a protein on mRNA, the first codon will start with AUG. All right? And I will go over that in more detail in 30B. You might also, some other features to notice on here, is that there's these weird areas on the table that just have the phrase TER on there. So here's another feature of the genetic code. There are only 61 of the 64 combinations, 61 codons that actually code for an amino acid sequence. So not all 64, only 61 that actually code for an amino acid sequence. 3 of them, labeled here, are called stop codons. So it turns out, again, you'll learn more in detail in 30B, that in translation, you need to stop translation at some point. But how does the ribosome know when to stop? You will find that it will stop when it reads one of the 3 potential stop codons. So these don't actually code for anything, except for one very special exception we'll talk about later. But these 3 are your stop codons. They normally will stop translation in the ribosome. All right. Vocab terms, triplet code, continuous code, non-overlapping code, degenerate code. Let's see if there's anything I've missed. Let me go back to this slide here. Let's see. So codons, yeah, you don't have to know this. This is history of how we were able to figure out all that stuff. Don't worry about that slide. Again, more history stuff. Don't worry about that. Oh yeah, you don't have to know the names of the stop codons. These are historical names for the stop codons. You don't have to worry about that. Let's see, we talked about 61 codons that actually code for an amino acid. 3, oh yeah, another phrase of stop codon is nonsense. It does not code for anything. Therefore, the ones that actually code for amino acid sequences are sense codons, so you'll find it very annoying that scientists like to reuse the same word in different contexts. In translation, sense means codes for protein, nonsense means stop codon. Yes?
Doesn't the AUG stand for the start?
Yes, that is the start codon, exactly, and which will be covered, I think, in more detail in chapter 30B, but you're exactly right. AUG is the start codon. Other vocab terms. Unambiguous. So, sometimes people get the phrase degenerate and unambiguous mixed up, so let me go back to the table to describe unambiguous. Let me make sure. Yeah. The phrase degenerate, again, means for one particular amino acid, you can get multiple codons that code for it. The phrase unambiguous means each one of these codons, and you look at each codon individually, they will only be able to code for a maximum of one amino acid. Okay, so be very careful. Unambiguous code means every time I see UAU, it will only code for tyrosine and no other amino acid. Every time I see CGG, it will unambiguously only code for arginine and no other amino acid. The phrase degenerate code means arginine has 4 different codons that code it. The phrase unambiguous means when you look at each codon individually, they can only code for one amino acid at a time. It's unambiguous code. One codon, each codon individually only matches to one of the 20 amino acids. All right. Other vocab terms here. Let's see. Degenerate. Okay, this is sometimes a memorization fact, so I don't really have any way of memorizing. Oh, actually, let me go over synonymous real quick. So, synonymous codons, so this isn't really applied to the whole genetic code. This is like a secondary vocab term. Synonymous codons are 2 codons that end up coding for the same or similar amino acids. So, let me see if I can find an example. Like AUU and CUG would be synonymous codons because they both code for what category of amino acid? What are isoleucine and leucine? Anyone remember the 4 categories of amino acids? What are isoleucine and leucine? Nonpolar. So the codons that code for 2 nonpolar amino acids would be synonymous codons. Although the context isn't really clear right now, there's a reason why we care about synonymous or non-synonymous codons, is that a lot of diseases arise based off mutations in the genetic code. But some of these mutations aren't as harmful as others. Here's why. If I have a mutation in my genetic code that caused it to change at the mRNA level, that caused it to change from CUU to CUC, has really anything changed at the protein level? No, because according to this table, that mutation caused me to make I used to make leucine, I am still making leucine with that mutation, okay? So that's fine, but that's not as harmful. There's another level of mutation, let's say maybe a mutation, this is a crazy one, that goes from CUG to AUU. This turns your protein at that spot, instead of making leucine amino acid, it's making isoleucine amino acid. But remember that with proteins, you learned way back in exam 1 that amino acid sequences interact with each other based off their chemical properties. So if I go from one nonpolar amino acid to another nonpolar amino acid, that's generally okay, not always, that's generally okay because it's still going to act as a nonpolar amino acid. So those are synonymous codons, and we really care about that in studies of mutations from one codon to another to see what changes at the amino acid sequence level occur. An example of a really bad mutation would be, let me find, where's the aspartic acid? Here's aspartic acid, and where's arginine? Would be if you had a mutation that turned GAU into AGA. You're going from aspartic acid amino acid into, does anyone remember what arginine is? What category of amino acid it is? It is a basic amino acid. That mutation is likely to be very serious because you're completely changing the chemical property of that amino acid. It isn't nonpolar, it's now acidic to basic, yes?
So synonymous means the ones that code for the same amino acid and if they're in the same category?
Annoyingly like that, yes. Synonymous is both same and similar amino acid, yes. Again, that is maybe more a memorization thing rather than having to know the specific details. If you see that on the exam, it will be simply memorization. I'm trying to give you a context of why that phrase even exists, and that has to do with those example mutations that I gave there. Another very annoying, very specific memorization thing you need to know about are these things, is that if the second base in a codon is a pyrimidine, which you need to remember are your C's or U's when we talk about mRNA, it usually, but not always, it usually codes for a nonpolar amino acid. It'll simply be a multiple choice answer. Second base pyrimidine usually codes for what? Nonpolar amino acid. Or the reverse. Nonpolar amino acid is usually indicated by what in the codon? Second base is a pyrimidine. Okay, so just memorize these facts, is that usually in your codon, which is a 3-base thing, if the second base is a pyrimidine, it's usually nonpolar. If the second base is a purine, it's usually polar. And then you might be wondering, well, what's the pattern for acidics? What's the patterns for basics? I don't know. We aren't given that information, so I don't care. So, moving right along. Actually, any questions before I go over this next concept? All right, so you learned a whole bunch of vocab terms just then associated with the genetic code. Make sure you know what each one means, what the context, what triplet code means, degenerate code means, all of that. All right, so I've already given the word start codon. You will learn in chapter 38 that that's where translation begins. The start codon is not always the very first 3 bases in an mRNA sequence. Okay, it is not always the very first 3 bases in an mRNA sequence. What this implies is that there's a whole set of bases before the start codon on the mRNA that completely get ignored. That region of mRNA that contains a bunch of bases that won't be translated is called the 5' untranslated region. The region of mRNA in the front that will not be translated. That is called the 5' untranslated region. The region that is actually coded on the mRNA is identified by this vocab term, open reading frame, or ORF. The open reading frame is the spot on mRNA between the start codon and the very next stop codon. mRNA can have multiple stop codons, by the way, but the open reading frame is between the start codon and the very next stop codon. Okay, there might be like 5 other stop codons after that. You don't count those. It's the start codon and the very next stop codon. So that is what the vocab term open reading frame means. Although it mentions this in bacteria, you can do this in any organism. You can scan mRNA sequences to find start and stop codons, because we know the pattern of a start codon is AUG, and we know the 3 possible patterns of a stop codon, so we can predict what amino acid sequences will form from mRNA, even if it's a brand new mRNA we've never seen before. And in the future, we may even be able to predict what protein forms. Once we know what amino acid sequence forms, right now our computers are not powerful enough to figure out how it'll fold and what 3-dimensional shape that amino acid sequence will form. So we can't predict what it'll do. If there's a brand new mRNA and a brand new amino acid sequence, we can't predict what it'll do. But in the future, we'll be able to predict functions of proteins based off simply the open reading frame that is given. All right, more vocab terms. Oh yeah, universal. Universal genetic code, this one's easy. That codon table applies to every organism that exists. Prokaryotes, eukaryotes, archaea, specifically fungi, animal, plants, all use the same table, the same code. That is what we mean by universal code. Of course, with every blanket statement, there's a few exceptions. I believe the 2 exceptions are listed right here in the next 2 bullet points. Okay, the 2 that you need to know about. So, out of the entire 64 codons on that table, there's one right here that is a little special, is the codon UGA. You go back to that table, and I will go back to it to illustrate this. You go find where UGA is, and you see normally, 99% of the time, it'll be a termination, it'll be a stop codon. It normally doesn't code for any amino acids, and it'll stop translation. However, sometimes, in mitochondria, if this codon is found, it actually codes for an amino acid, in this case, tryptophan. Okay? And one of the theories why this might happen is right here in purple, is that we know the ancient symbiosis hypothesis is that the very first eukaryote came into form because it absorbed a prokaryote, which eventually would become mitochondria. So mitochondria has all these weird properties. We learned last time mitochondria has circular DNA, despite being located in a eukaryote. Here's another cool difference here, is that if you look at mitochondria, the codon UGA actually codes for tryptophan instead. Other differences, let's see, it's still with UGA, yeah. So, remember I keep saying 20 common amino acids, 20 common amino acids. Oh yes, question.
So before, like, you said methionine only has like 1 codon.
Yes.
On the slide, I think it said that tryptophan also does, but does it technically not, since of the...
I must have missed one, yeah. I always go with methionine because that's the start codon. But yeah, most of the time you can see that tryptophan only has 1 codon. However, now we were given that one exception that UGA can also code for tryptophan. But you won't be asked what are the amino acids only coded by 1 codon. You may be asked what's the start codon though, which is AUG. So back to those exceptions. Again, the one exception we have so far is that UGA in mitochondria will code for tryptophan. Another exception has to do with a unique amino acid. Remember that there are 20 common amino acids. There's a handful of special amino acids, one of which is called selenocysteine. Okay? Selenocysteine, and it's stated on here. You won't have to know what it looks like. Selenocysteine is an analog of cysteine, and it is coded by UGA. So UGA looks like has its own weird rules associated with it. Sometimes it's a stop codon, sometimes it codes for tryptophan in mitochondria, and sometimes it codes for something called selenocysteine in some prokaryotes and eukaryotes. So, let's see. So you might wonder, what amino acid is selenocysteine made from? And if you said cysteine, you're wrong. For whatever reason, and I still don't know this because I don't bother looking at the structures of these things, it turns out selenocysteine is actually derived from serine, okay? And that's stated here, is that selenocysteine comes from serine, which is on its tRNA like normal, but that serine, if you remember the structure of serine, it has a hydroxyl group at the end of it. It'll be modified and transformed into selenocysteine. So a couple weird phrases on here, and I'll try to break it down to you as different concepts. One, selenocysteine is a special amino acid coded by UGA. Okay. Two, selenocysteine, although is an analog of cysteine, confusingly, it is made from serine. It's made from modified serine. Okay, so try to keep those facts distinct, but they are all technically true things. I'm not a selenocysteine expert. I don't do research on this. So I don't know why that is. But again, selenocysteine is an analog of cysteine, but physically when it is made from serine. And we'll stop for now, and we'll continue with 30A and 30B, which will not take that long. All right, I'll stick around if anyone has any questions.